Okay, it's demo time. Exactly how much time do I have? Are we going to noon? Okay, so my name is Rob. I'm a graduate student, a very, very, very senior graduate student in the Nolan Lab. And so what I'm going to show you today really quickly is how to use SPADE in Cytobank, if that's what you're interested in using to analyze your data. And then if we have a little bit of time at the end, I'll get to some additional work that I've been doing on testing or developing methods that allow you to test specific hypotheses within your data. So quickly to SPADE, you've seen this many, many, many times before. As Federico mentioned, there are many ways for you to get access to SPADE. It's an R package. You can use it by calling SPADE from this Cytoscape software package. And then finally, there's this web interface that Zach Bjornsson, who is a graduate student in our lab, has been working with Cytobank to implement so that you can call it directly after uploading your data to Cytobank. And that's what I'm going to show you today. Federico already covered this, and I'm sure other people have, so I'm just sort of going to skip through it. The most important, what I'm going to show you here is briefly what happens with all of your data during this process, simply so that I can show you what you need to tell, how you have to configure SPADE in order to run it. So you'll start with all of your data in an FCS, in these separate FCS files. Let's say these represent different patients, or maybe this is the same patient that is stimmed with different experimental stimuli or different drugs. And so what SPADE is going to do is it's going to take all of this data and combine it together. Or sorry, it's going to perform its density-dependent downsampling first, and then it's going to combine all of this data together into one single big clump of data. It will cluster that data together, and then it splits it back out into the original files from which all the data came, upsamples, like Federico mentioned. And the end product of this is you have your clustered FCS files that it will then display in a tree. So again, this is kind of the workflow. The things that are highlighted in red here are the things that you have to configure when you run a SPADE analysis. So one of the things you have to tell it is, how much do you want to downsample? How much do you want to get rid of those extremely abundant events that you've measured? Maybe you have lots and lots of T cells that you want to get rid of, so you set a low density downsampling target to make sure you get rid of lots and lots of overrepresented events. From what I've looked at, this number doesn't seem to make a huge impact on the results that you get from the SPADE tree. So I wouldn't lose too much sleep over setting this parameter. There's a default set in Cytobank, and you can probably leave it at that unless you have some good reason to want to set it otherwise. The other thing you have to do is tell it which parameters to use for clustering. Now, typically, as Federico mentioned, when you perform clustering, we generally do this using markers that are typically used for gating. So clustering is just sort of it's an analogous process to gating. We want to find all the cell subsets that have similar expression of cell surface markers. So when you're telling SPADE what markers to cluster on, we usually just tell it to cluster using cell surface markers. The other thing you can tell SPADE is, how many of these little clusters do you want to discover? Now, again, the results don't seem to matter too much based on you changing this number. What you're really just trying to do is partition the cells up into enough small cells that you're successfully able to isolate those rare events that you might be interested in finding. And finally, once you've set this, the only other thing you need to tell it is, what are the reference conditions? If you've measured multiple experimental conditions that you want to compare, you need to say, this is my unstimulated group, these are my stimulated cells, so that it knows how to calculate the fold changes and compare those between your different groups. So that's the basic workflow. Now I'm going to show you a demo on dvs.cytobank.org. There is a publicly available experiment that says SPADE demo. If you want to recreate this analysis that I'm about to show you or follow along in your laptop, you can do that if you have a DVS Cytobank account. What I'm going to show you today is just a very simple example using PBMCs measured using fluorescence flow cytometry. It's got 4 surface markers, 2 phosphoproteins, And these data were measured under these 4 different experimental stimulation conditions. Okay, so here's DVS Cytobank. And here's, can you guys read this in the back? Yeah. No, yeah, okay. So here's the demo experiment. Again, anybody can actually make a copy of this experiment and run this analysis if you're interested. So here are the list of files that I have. I have these PBMCs that are measured, one that's been stimulated with IL-10, one with IL-6, one with LPS, and then I've measured 2 samples in an unstimulated state with no experimental stimulation. And so I can start by, over here there's this sidebar that says SPADE, and I can click on Create SPADE Analysis, and I'll just call this a test demo. And so this is where I need to configure those parameters that I was talking about in the slide beforehand. So here's where you can set the target number of nodes that you're going to be clustering towards. This is where you can set the number or the downsampling target percentage. They're kind of over here. I wouldn't worry too much about setting those or changing those parameters. These are usually the things you need to select it in order to run. So first thing we're going to do is select the clustering channels. So you can click Choose. And in this case, we're going to use the surface markers, CD3, CD20, CD33, CD20, CD3, and CD4. We're basically going to find cells that express similar amounts of these cluster surface markers. And we'll say Done. And the other thing we have to do in this circumstance is, what I would like to do with these data are see the difference in signaling between my unstimulated samples and my stimulated samples. So I'm going to choose this. I'm going to select the different groups for fold change references. So it's going to ask me, what are my reference files? What's my basal condition that I'm interested in comparing against? So I'm going to check the 2 unstimulated samples here, because these are the reference conditions that I will want to evaluate signaling in response to. So I'm interested in seeing what the signaling does in these stimulated samples versus these reference samples. So I've selected those. And the thing that I'm not going to do here is I've already actually gated these data out for singlet gating, so you can run SPADE on a subset of your data that you've already gated. Let's say you have a particular branch of, you're only interested in looking at T cells with SPADE. So you can run SPADE just on a particular subset of, or a particular population that you've already gated. I'm just going to go ahead and run it on everything. So that's basically it. So we've set our parameters. And once you're done, you click Start Analysis, and you'll see this bar that goes off and it starts turning green. And eventually you'll get an email that says, hey, you're done. And then you can look at the results. So I've gone ahead and I've already run this analysis, this exact same analysis that I showed you how to configure just for the interest of time. So here's what the results look like. So what you'll get back is this tree, a tree, that you've been seeing a lot of. I will, let's say, to make this a little bit easier to see, I'll turn this. No, that's not making it easier to see. OK, so I'll go ahead and we'll use this color scheme. And what you're mostly interested in here are actually starting to annotate this tree like Federico described earlier. We have a bunch of clusters that are connected together, and you use the coloring information. You color these nodes with different parameters to help you annotate the tree. So the first thing I might be interested in doing is looking at the expression of CD3 in these nodes. And whoo, can you tell that these guys are red? Is that, yeah, OK. So these nodes and these nodes all express CD3, so that tells me something. Maybe I look at CD4 next, and I see that these nodes are also very red, and that means these guys express high levels of CD4. So because these express high levels of CD3 and CD4, I'm just going to go ahead and assume that they are CD4 T cells. And so I can select those guys together and make an annotation bubble. Down here, there's an annotation panel. and I can say add bubble. And it'll ask me what those cells are, and I'll just call them CD4 T cells. So now I have an annotation on my tree that helps me, as I continue down the road in my analysis, look at these and consider that all of those guys are CD4 T cells. I can do that with some other things just for fun. Let's look at CD33 expression. I see that this guy down here, this branch is highly populated by CD33. So I'll select those, and I'll add another bubble. We'll call these monocytes. OK, so there we have some cells. And if you're interested, the tree is basically a way for you to explore your data in this tree form. But you can do a lot of the same things that you would do using regular flow analysis. So if I just select these CD4 T cells, this particular cluster that I'm looking at, I can look at the scatter plots of those markers. So if I plot CD3 by CD4, what this plot here shows you is the scatter plot of all the cells that are selected in these nodes. And then in gray, in these little gray background contour lines, those are all the other cells. So I can see that this population of cells, this CD4 T cell population that I've defined, has high levels of CD4 and high levels of CD3. It's just another way for you to confirm that what you've selected, it makes sense and is rational. So that's one way to annotate things. Now we might want to go on to a different, we want to see what these guys are doing. So let's start coloring by some of our functional markers. So we can color by phospho-P38. In our unstimulated state, phospho-P38 isn't doing much. So red, according to our scale here, red is high, but we don't really see a lot of red on the board. So what we can do is, instead of just looking at the mean, let's look at the fold change. And this is where we bring in our fold change versus our baseline condition. So we wanted to say, let's compare the levels of these things relative to our unstimulated state. But instead of looking in the unstimulated files, let's look at these things in our LPS stimulated sample. So here's LPS stimulation. We're looking at the levels of phospho-P38, and we're comparing the fold change versus baseline. And so when we color by that, what we can see is that the monocytes really light up. So we see that there's a functional response in this branch of the cells that are telling us that these guys are changing significantly in response to our stimulation versus our unstimulated baseline. This is the same kind of visualization that you would see using a heat map, but this just shows it in nice spade tree form. So we can do something else. We can say, let's look at STAT3 response, not really going not a lot of STAT3 response in LPS stimulated cells. But if we look at IL-6 stimulation, we can see that in our IL-6 conditions, the CD4 T cells have very high expression of STAT3 relative to our unstimulated baseline. And so this is sort of how you move through the tree and get an idea of what the functional activity of these cells are in the different trees. There are a lot of different things you can change with the coloring parameters, but this is the basic overview of how you annotate a tree and sort of explore the functional behavior in your measured data. So I think in the interest of time, I'm going to stop here on this particular subject. Do you guys have any questions about using this interface, what you can show, how to sort of represent different pieces of information that you might be interested in asking? OK, you can always ask at a later point, too, if you have questions. OK, so I'm going to close that. And OK, so now we're going to switch gears a little bit. I have 20 minutes left, or a little bit less. So the work that Federico has been showing you and the work that's done with Spade, a lot of these things are sort of focused on visualizing the data, allowing you to explore your data, helping you develop an intuition about what is sort of going on in different cell subsets. I'm going to be talking about something a little bit more quantitative, in that a lot of times when you do these experiments, you come in with prior hypotheses about what you think are going on in your data, and you want to test to see if those hypotheses are true in the data that you just measured. the work that I'm going to show you right now is a method for allowing you to test those hypotheses. So what kind of hypotheses am I talking about? Let's say you measure a bunch of patients with AML, and you measure a bunch of healthy reference patients, and you have this hypothesis that there are some cell subsets in your AML patients that exist that do not exist in your reference data sets or in your healthy reference samples, and you want to see if that is true. Can you find the cells that exist in your AML patients that do not exist in your healthy reference? Maybe you have a bunch of AML patients and you have some clinical outcome that you've measured, let's say relapse-free survival time, and you want to ask the question or test the question, are there any cell subsets that are predictive of time to relapse in your AML patients? These are the kind of hypotheses that we're talking about. Finding associations between the data that you've measured and some external experimental endpoint that you are interested in testing for an association with. So there are many examples of this. Maybe one of the most famous is a detection of an association between CD4 T cell counts and HIV development. You might be interested in looking at MRD in leukemia patients. One example that came out of our lab was Jonathan Irish showed that if you looked at the signaling in a subset of B cells in follicular lymphoma patients, patients that had a very large percentage of B cells that did not signal correctly after stimulation, these patients had much poorer clinical survival than patients who had B cells that did signal correctly after stimulation. So the objective here is really to find these cell subsets that tell us something about this external question we have. We want to find these cell subsets that tell us about how our patients are going to do, our patients' phenotypes. And really, often the experimental challenge is for you to identify those cells in the first place. When I tell you about the CD4 T cells or MRD or these B cells, the experimentalist had to do a lot of work in order to find what those cells were in the first place. And once they found them, then the interesting biology started to happen. So the work that I'm doing is really trying to help you answer the question, can you automatically find these cell subsets that will tell you something about your experimental endpoint of interest? So I have a method termed Citrus. I'm from Florida. It's an homage to my home state. Basically, it allows you to help you find cell subsets of interest that are associated with an experimental endpoint of interest that you might have. This is what I sort of view as the traditional manual analysis workflow for performing this kind of analysis is. You basically start by measuring your data by flow cytometry. You manually gauge your data, and then you quantify some effect of that about you quantify some property of the cell subsets that you've determined. What are the abundance? What's the abundance of this cell subset that I've identified in each of my patients? And then you test to see if that feature that you've quantified is somehow predictive of your clinical endpoint of interest, or you say like a t-test to say, is the abundance between these two patient groups different in this cell subset? And that's really sort of how you arrive upon that cell subset that you're interested in exploring further. So what Citrus does is it mimics this process, but it just replaces it with some intelligent computing methods that make it a little bit quicker and easier. So instead of manually gating our data, we're going to use hierarchical clustering to find all the different cell subsets that we can in the data. And then we'll go through, and then we basically quantify features of all the different cell subsets that we can find in our data. And then we plug these features into a regularized supervised learning algorithm. So that's a mouthful, but all you really need to know is that given a bunch of cluster properties that might be interesting, and an external question of interest, say finding the difference between healthy patients and diseased patients, the regularized algorithm will dig through all of the data that you feed it and give you back the things that are informative with respect to your endpoint of interest. It'll say, you gave me 100 cell subsets. I'm telling you that these two are the ones whose abundance differs between your healthy and diseased patients. And so we can evaluate this model using some cross-validation techniques. But really what it comes down to is if the model can find something that's strongly associated with your endpoint of interest, those are in fact the cell subsets that you're interested in finding in the first place. So it's kind of it's a slightly roundabout way of getting the existing, or performing the same process that you would by hand, except we now have computational methods to make this more efficient, especially in circumstances where you have data measured with CyTOF and you can't really explore that data entirely by hand. So that's the general overview. To try and make this more concrete, I'm going to I'll show you two very trivial examples. I don't think I'll have time to demo the software for you now, but if this sort of thing interests you, I'd be happy to show you the software. It's available as a free R package, and it has a web interface. So we'll get to that. We can talk about that later in lab. Okay, so the two examples that I'm going to show you, one is just a very simple, trivial synthetic data set that I've generated just for the purposes of illustrating how the method works. In this case, we have a bunch of healthy patients and a bunch of diseased patients. We have data that's measured in just 2 dimensions, and the healthy patients has lots of cells up here and not a lot of cells here, and that's flipped in our diseased patients. In the other example I'll show you in just a second, we're going to use this method to identify cell subsets that respond to experimental stimuli in blood. Just very simple example, trivial example case, and then trivial biological example. But you can use your imagination and figure out how this would sort of generalize to the question that you might be interested in asking with your data. So here, again, now I'll show you the synthetic example. So we have a bunch of healthy patients, a bunch of diseased patients. We have their 2-dimensional data. 10% of their cells in the diseased patients are up here. 30% of their cells are in this bottom population in the diseased patients. And again, that's flipped between our healthy and diseased patients. There's noise in this, but that's not really the point. We take all of the data from all of the samples, and it gets pooled together, and we cluster that combined data so that we can find clusters that we can look at in each of our individual patients. So we'll take the combined clustered data, and we'll split it back out into the individual patients, and then I can start to quantify properties of each of these clusters in each individual patient. So for example, we can say, what is the abundance of this red cluster in patient 1? How many of their cells do they have in this cluster? How many of their cells do they have in blue? How many of their cells are in cluster green? And so we'll do that for patient 1, and we'll do that for patient 2, and we'll do that for all of our patients. So we're quantifying properties of the cell subsets that we've discovered in the data using hierarchical clustering. After that, we'll take this cluster, this matrix of cluster properties. We know which of these patients are healthy and diseased. We know that already based on our external experimental information, and we'll pass it into this regression algorithm. And we ask the regression algorithm to find us the subsets of these clusters that differ between our healthy and our diseased patients. And so the model does some internal evaluation of the value of each of the features, how predictive are they, and what it spits out are these set of features. So it'll say something like this. It'll say cluster 99993, which is just an arbitrary number. The abundance of that cluster is very high in healthy patients and very low in diseased patients. And so each dot here represents a healthy patient, as I specified, and each dot here represents a diseased patient. And we can see, yes, that the healthy patients have lots of cells in cluster 99993, and the diseased patients do not have a lot of those cells. And in cluster 99994, it's split, or it's switched. Healthy patients don't have a lot of those cells. Diseased patients do have a lot of those cells. And so once we know that this cluster is informative, 99993 and 99994, once we know that those are interesting, then we just use standard scatter plots or density plots to figure out what that cluster is. So we plot the markers that were used to identify that cluster. And so these are those clusters. So this is a plot of cluster 99994. This is a plot of cluster 99993. And just to sort of bring it full circle, as you recall, these were the populations that we had originally said were different in our synthetic data. So this is how we go from taking a data set where we don't really know anything about it and identifying the features in it that are predictive of, say, a sample group or a clinical outcome, and do that in an unsupervised manner without any hand gating, and so on and so forth. So, yes?
So what happens if in your disease group 50% of the population is responding one way and the other? In your original part where you combine all the data, then are you going to lose that data?
To clarify, are you asking what happens if you have subtypes of a particular disease? Okay, so that's something that's extremely difficult to automatically detect. What that will look like when you do this is that some of these values that you have will be overlapping, right? You know, this is a very trivial example where I made it easy for these things to be different simply because I just wanted to show how it works. In your case, those values of those different groups would be, you know, more overlapping. And all you can really say from that is that the feature that this method identified is useful for some of your patients, but not for all of them. This method is not explicitly trying to identify what are the subtypes of your diseases that exist in this. It's just out of the beyond pay grade. Yeah.
But the 2 slides back when you showed the first step where you're pooling all of your patients' data together into one data set. So those sort of nodes that would come out as unique won't come out as unique if there's heterogeneity in your disease set, right? So you won't identify those areas to chase, is that right?
Well, because you're combining them all together, it's looking over the space of all the potential clusters in all of your samples. So if there is one particular cluster that exists in a subset of your patients, it will consider that cell subset across all of your patients. It just won't, that one particular cell subset that it identifies just won't be a great predictor for all of your patients. Okay, so, oops. So that was a very trivial, yes, sorry.
Sorry, I maybe I don't understand something, but if you tell it which are healthy and which are diseased, doesn't that sort of make it supervised? Like if you tell it to tell you which are which, can it do that?
It is supervised. So this is specifically for the circumstance where you come in and you have some sort of external information about which patients belong to which groups. You have a bunch of patients that respond to chemotherapy. You have a bunch of patients that don't respond to chemotherapy. That's your finding. You want to know what's different between them. This isn't designed to discover subtypes within a particular set of measured samples. It's only designed to test for specific hypotheses that you have based on external information. Does that kind of get at your question?
I thought you said unsupervised.
Sorry,
I
did. So the discovery of the cell subsets is unsupervised, whereas Federico's approach uses this prior understanding of cell types that exist in your data to help guide the layout of the tree, at least initially. This makes no assumptions about what cell types exist in your data, which can be good and bad. It can be good because you won't be biased in looking at things that you know exist, and it can find things that you didn't know were important. It's bad because there's an issue of multiple hypothesis testing, and it's bad because we ignore 2 decades of prior immunological research. But, you know, balance those. So very quickly, let me show you what this looks like in practice in a real data set. So Eli showed you one slide from his barcoding work earlier. They took a bunch of patients' blood and measured PBMCs under different experimental stimulation conditions. And so one of those experiments that he performed was looking at responses to things like a BCR crosslinker. And so we went back and mined that data just to ask the very simple question, given a bunch of unstimulated samples and given a bunch of PBMCs that are stimulated with B cell receptor crosslinker, what are the cell types that respond in terms of their functional markers to this B cell receptor crosslinker relative to this unstimulated baseline? So my 2 groups are BCR stimulated and unstimulated. The question is, what are the different things? Very simple, but again, for proof of concept, I'll tell you right now, the thing that we find is that's the best predictor between these two groups is the activation of phosphoproteins in B cells, which is not very exciting, but is a nice confirmation that it gives you a sane answer. But it also identifies a bunch of other stuff that we weren't expecting to see. Some of those things are real, some of those things are not. So this is what that experiment looks like. Again, they had a bunch of healthy patients. They took aliquots from each of the healthy patients and stimulated them with B cell receptor crosslinker. Also measured a bunch of unstimulated samples, measured it by mass cytometry, and we plugged it into our algorithm Citrus. They measured a standard barrage of immune lineage markers and a bunch of functional markers that basically quantify the activity of different pathways that are downstream from various numbers of receptors. We ran our method. So what does it give out? So the method, we ran this. We asked it to find us differences between our different stimulated groups. The methods spits out this list, this ordered list of things that are different between our two groups. And the feature that showed up at the top was that the level of phosphorylated S6 levels was extremely high in B cell receptor cross-linked patients and extremely low in reference patients in this cluster, 75561. So what is cluster 75561? If we plot these histograms where, can you tell that this is red? Okay, so the red lines show the abundance of the surface markers in cluster 75561, the important one, and black shows everything else. This black dotted line shows everything else. So we can see that cluster 75561 has relatively high levels of CD20, has relatively high levels of IgM, HLA-DR. It has relatively low levels of CD7 and CD3. And so you show this to an immunologist, and they say, great, you found that S6 signaling in B cells tells you the difference between samples that were stimulated with B cell receptor crosslinker and B cell and unstimulated samples. And again, while that's not mind-blowing, the algorithm had no idea what B cells were when we plugged in this data. We just dumped the data in. We said, find me cells types that are different between stimulated and unstimulated samples. And the first thing that it spits out is kind of the thing that you would expect. So that at least what I'm trying to do is convince you that this might work on this very trivial data, so it might work on your really interesting experimental data as well. So we did actually end up go in and validate a lot of these responses to confirm that they were actually indeed biologically induced. But then there were a bunch of other things that showed up in addition to B cell responses. We had levels of S6 responding in monocytes. Those turned out to be true as well. And then a bunch of responses in things like CD4 T cells and other unknown cell types, things we didn't have any idea what these cell types were. These things turned out to be experimental batch effects, like that were just induced by particular protocols that were used to stain the cells. There were systematic variations in the way some of these things were stained on a particular day. And so we get these responses in cell types that we wouldn't expect. Now, the reason I'm pointing this out to you is not because batch effects are interesting to find, but just that using an unsupervised method like this is going to tell you all the things that differ, or all the things that are associated with your question, your experimental endpoint of interest. And you have to go in and convince yourself that some of these things are real and interesting, and other things are perhaps experimental batch effects. And you have to do the legwork of figuring out what is biologically meaningful and what is not biologically interesting. OK, so if you just want to ignore everything else and just download the code, it's right there, and go to town. I will, in my last 3 minutes, show you very quickly what that analysis that I just showed you looks like from a software perspective. It's an R package, but it comes with a graphical interface. So all you have to do is select one of the FCS files that you're interested in looking at. So here are all the stimulated and unstimulated FCS files that I analyzed in that last example. And it will pull, after you select that, it will pull up a web interface. And here are the different samples that you're looking at. You can say, one of my groups is BCR stimulated. And it will go ahead and select all the samples that have BCR in the title. The other group is unstimulated. Unstim. It will select those guys. So this is where we're sort of framing our question of interest. Saying which samples belong to which groups based on your prior biological hypotheses. We have to simply specify what the clusters or what the markers are that we want to use to cluster. Again, these are generally cell surface markers, your CD markers. We can tell Citrus to calculate different properties of those clusters. Maybe you're interested in looking at abundance changes, or maybe you're interested in looking at median functional markers. So for this analysis, I would say that I'm interested in looking at cluster medians and select all the different phosphoproteins that I think may be responding to my stimuli. And then finally, there are a variety of statistical models that you can use to detect for the differences between these two groups. I'm not going to describe them here, but I will just say some of those are useful for identifying associations with continuous endpoints or survival time, whereas other ones are just mainly focused on detecting differences between two or more groups. And then you hit the run button, and it creates a bunch of output for you, but I do not have time to go through each of the individual outputs. But that's the overview of the software. It's available on GitHub. There are some installation instructions, and I'm also happy as much as I can to try and work with you to try and get it working on your data as well. And so I'll stop there and take any questions. Yes?
Can you talk a little more about normalization? If you have enough variation day to day, can you just not actually be able to detect real differences as well, or are you just going to pick up artifacts?
So that sort of depends on the variation how the variation is manifested in the way you are in the effect that you're trying to quantify. If you have lots of variation in your staining of your surface markers, when you perform the clustering, things that are CD4 T cells in one patient might appear sort of in a different multidimensional space. I haven't been able to characterize this fully in a statistical manner. My current intuition is that when you're working with things in very high dimensional space, that day-to-day variation actually doesn't make a huge difference in the way things cluster, because the cell types that you're identifying, the clusters that you're identifying, are so unique in this high dimensional space that a little bit of variation in the way these things are measured doesn't make it very close to another cell type accidentally. That's at least my current sort of intuition. If you're working with, say, 2-dimensional data or 3-dimensional data, then absolutely it makes a difference, and you have to sort of scale the data so that those distributions line up between your measured markers. In terms of quantifying an effect such as a phosphoprotein signaling level, absolutely. If you have huge amount of variability in these signals, that gives you huge inter-patient or intergroup variability, and you're going to need more patients to detect that. I don't know of any great way to normalize that without introducing some very large biases into the data. So it's best if you can sort of normalize that ahead of time using software such as the B normalization software that Rachel has developed. Did that get covered? No? Okay. There are normalization pieces of software that I'm sure we can provide that help you hammer out some of that intra-patient variability. Other questions? Yes? Yes. Yeah, so the way that the particular methods that this uses are non-parametric, and we don't control by p-values, we just control by false discovery rates. So, but yes, the list that you get out is ordered by the most significant results to the least significant results. And so you have to sort of make the decision when you're doing this analysis that you want to be very inclusive with your results, or very, very restrictive, so that you have high confidence that the things you're reporting are in fact real. Okay.